In this video lecture, I'll be talking on transplant immunology. And we mean by transplant immunology, immune responses to grafted organ or tissue. Accordingly, the donor and recipient are matched for immune compatibility before transplantation. Transplant types. There are 4 types of transplant, and these are autograft transplant, which is within the same individual, so no immune response. Isograft transplant or plant between two identical twins, known as syngraft. The histocompatibility antigens are identical in donor and recipient. Allograft, it is between two individuals of the same species. But however, allograft displays specificity and memory. That is to say, alloantigens may elicit both humoral and cell-mediated immune responses. The rate of allograft rejection varies according to the tissue involved. For example, skin grafts are rejected faster than other tissues, such as kidney or heart. In skin graft, B cell, T cells, and natural killer cells may lead to rejection of the graft. So, allo-specific antibodies may reject the graft through complement activation, and this one of the characteristics of the specificity. The fourth type of transplant is xenograft, and it is between animal and human of different, so they are of different species. Transplanted tissue may be part of liver, heart, kidney, hematopoietic stem cell, skin, corneas, pancreatic islet cell tissues. Before any tissue transplant, some tests should be performed to avoid rejection and complication. And these include ABO antigens compatibility, so the donor and the recipient should have the same blood group. Since differences in blood group are responsible for the most intense graft rejection reaction. The blood group antigens are expressed on red blood cells, epithelial cells, and endothelial cells. So antibodies produced in the recipient to any of these antigens that are present on transplanted tissue will induce antibody-mediated complement lysis of the incompatible donor cells. Also, tissue typing. It is very important to type the tissues between donor and recipient before transplantation. But however, I just would like to give an account on HLA compatibility before talking about tissue typing. HLA, there are two classes actually. here in the field of transplantation, human leukocyte antigen class 1, and this is expressed on nearly all nucleated cells. Human leukocyte antigen class 2, this expressed on the surface of antigen presenting cells, which are professional antigen presenting cells, and these cells are B cells, dendritic cells, and macrophages. HLA molecules are encoded by cluster of genes within the chromosome number 6. HLA class 1 molecules encoded by cluster of genes within 3 loci known as HLA-A, HLA-B, HLA-C. HLA class 2 molecules are encoded by cluster of genes within the loci HLA-DR, HLA-DQ, HLA-DB. So, and now I am going to talk about tissue typing or typing by using a polymerase chain reaction or PCR prior to transplantation. And this technique actually it is used in the field of transplantation for detection of HLA compatibility between donor and recipient, and we use PCR as it is useful to avoid an early rejection, especially the genes within HLA-A, HLA-B, HLA-DR. ELISA for typing. ELISA or enzyme-linked immunosorbent assay. It helps in avoiding rejection by antibodies. And as you may know that some individuals may upregulate antibodies to HLA, and this is following actually transfusion or in physiological process like pregnancy, as pregnant women to different may upregulate to different babies different HLA amount of antibodies against HLA, especially anti-human leukocyte antigen IgG antibodies. This may lead to rejection. ELISA actually also detect antibodies against genes within HLA-C loci and genes within HLA-DQ and DB. And also this technique is useful in the field of transplantation, which is MLR or mixed lymphocyte reaction, and it is actually lymphocyte cross match, and it is very useful for detection class 2 mismatch. Also, other tests are required before transplantation. For example, some tests to detect infections. For example, to detect cytomegalovirus, Epstein-Barr virus, herpes simplex virus, hepatitis B virus, hepatitis C virus, West Nile virus, and tuberculosis. So following transplantation, these are the possible complications: rejection, renal insufficiency, osteoporosis, cancer, infection due to immunosuppressant. Types of rejection. Actually, the most dangerous type of rejection is hyperacute, which is on-table rejection during performing the operation, and it is due to preformed antibodies that have the capacity to fix complement, and it takes actually place rapidly, as I mean on table or within the first 48 hours. Accelerated rejection, it is due to pre-existing of non-complement fixing antibodies, and it takes place within few days. Acute rejection takes place within days and maybe up to 6 months time, and mediated by T cells due to delayed hypersensitivity reaction. Chronic rejection takes place months following transplantation due to immunosuppressant, infection, renal insufficiency, fibrosis, ischemia. Fibrosis may result from harsh or difficult surgery. Graft versus host rejection, known as GVHR, and in this type of rejection, the graft starts to reject the whole body, and it is due to presence of T cell within the donor graft, which make the response against recipient antigens. I would like to thank Dr. Abir Abdelmunim for taking part in preparation to this lecture. Thank you very much, Professor Ahmed Bolad.